1. Q. What is molecular diagnostics in virology? A. Molecular diagnostics in virology involves the use of techniques such as PCR, sequencing, and microarrays to detect and identify viral nucleic acid in patient samples. 2. Q. What is PCR and how is it used in molecular diagnostics for virology? A. PCR, polymerase chain reaction, is a technique used to amplify a specific region of DNA or RNA. 2. In virology, PCR can be used to detect viral nucleic acid in patient samples. 3. Q. What is the difference between PCR and RT-PCR for viral detection? A. PCR is used to amplify DNA, while RT-PCR, reverse transcription PCR, is used to amplify RNA by first converting it to complementary DNA, cDNA. 4. Q. What is the importance of primer design in PCR for viral detection? A. Primers are designed to specifically bind to the target viral nucleic acid, allowing for specific amplification and detection. 5. Q. What is the difference between conventional PCR and quantitative PCR, qPCR, for viral detection? A. qPCR allows for quantification of the amount of viral nucleic acid present in a sample, while conventional PCR only detects the presence or absence of viral nucleic acid. 6. Q. What is the role of sequencing in molecular diagnostics for virology? A. Sequencing can be used to identify the specific strain of virus present in a patient sample, allowing for epidemiological tracking and identification of drug-resistant strains. 7. Q. How can microarrays be used in molecular diagnostics for virology? A. Microarrays can be used to simultaneously detect and identify multiple viral strains in a patient sample. 8. Q. What is the difference between targeted and untargeted sequencing in virology? A. Targeted sequencing involves sequencing a specific region of the viral genome, while untargeted sequencing involves sequencing the entire viral genome. 9. Q. How can next-generation sequencing, NGS, be used in molecular diagnostics for virology? A. NGS can be used to sequence entire viral genomes quickly and efficiently, allowing for identification of novel strains and characterization of viral diversity. 10. Q. What is the role of bioinformatics in molecular diagnostics for virology? A. Bioinformatics is used to analyze and interpret the large amount of sequencing data generated in virology research, allowing for identification of viral strains and characterization of viral evolution. 11. Q. What is the difference between in situ hybridization and PCR for viral detection? A. In situ hybridization involves detecting viral nucleic acid directly in tissue samples, while PCR amplifies viral nucleic acid for detection. 12. Q. How can multiplex PCR be used in molecular diagnostics for virology? A. Multiplex PCR involves amplifying multiple target regions simultaneously, allowing for detection and identification of multiple viral strains in a single sample. 13. Q. What is the role of reverse transcription in RT-PCR for viral detection? A. Reverse transcription is used to convert viral RNA into complementary DNA, cDNA, for detection by PCR, since PCR only amplifies DNA. 14. Q. What is the difference between real-time PCR and endpoint PCR for viral detection? A. Real-time PCR allows for detection of amplification in real time as it occurs, while endpoint PCR requires gel electrophoresis to visualize amplification after it has occurred. 14. Real-time PCR is faster and more quantitative than endpoint PCR. 15. Q. What is the role of probe design in molecular diagnostics for virology? A. Probes are designed to specifically hybridize to the target viral nucleic acid, allowing for specific detection and quantification. 15. Probes may be labeled with fluorescent or other reporter molecules for detection. 16. Q. What is the difference between sensitivity and specificity in molecular diagnostics for virology? A. Sensitivity refers to the ability of a test to correctly identify positive cases, while specificity refers to the ability of a test to correctly identify negative cases. 16. A test with high sensitivity and specificity is considered reliable and accurate. 17. Q. How can nested PCR be used in molecular diagnostics for virology? A. Nested PCR involves using two sets of PCR primers to amplify a smaller target region within a larger target region, allowing for increased sensitivity and specificity in detection. 18. Q. What is the difference between conventional PCR and real-time PCR in terms of detection limit? A. Real-time PCR has a lower detection limit than conventional PCR, as it allows for detection of amplification as it occurs in real time, rather than after the reaction has completed. 19. Q. How can droplet digital PCR, ddPCR, be used in viral detection? A. ddPCR partitions a sample into thousands of individual droplets, each of which undergoes PCR amplification. 19. The number of positive droplets is then used to calculate the absolute quantity of viral nucleic acid present in the original sample. 20. Q. What is the role of viral load testing in monitoring antiviral therapy? A. Viral load testing measures the amount of viral nucleic acid present in a patient sample, allowing for monitoring of the effectiveness of antiviral therapy. 20. Decreasing viral load indicates that the treatment is working, while increasing viral load may indicate drug resistance or treatment failure.